changing type. So you can see why that might be important. The next things are TUJ1 and TH. These are markers of midbrain and dopaminergic neurons, respectively. So this was how the researchers measured for the fibroblasts that were converted into neurons, since neurons express these things and fibroblasts don't. So in short, more TUJ1 and TH is better. So higher levels of those are what the researchers are looking for. And then the last thing is DAPI, which is a term simply meaning the total number of cells in that experiment. So if you see something like percent DAPI, it's a percentage of cells that express TUJ1 or TH, for example. And also, this will be important a little bit later on, but this is a little visual of the cell cycle if you need a quick refresher or may not know. The first stage is G1, S, G2, and then finally M. That will be important in a little bit. So the first thing the researchers did is basically what they already knew. They activated the A and L genes in fibroblast cells and see how many of them were converted into neurons. So this picture over here is an image of that, and so is this first bar right here graphically. Basically, in these types of graphs, more rainbow is better, and this did not make a lot of rainbow. They found that about 18% of fibroblasts were converted into neurons, which isn't great. It's not a very high rate of conversion just by activating the A and L genes alone. So they decided to look into more ways where they could increase the rates of conversion. The first thing they did was they reasoned that, well, if P53 keeps cells differentiated into a specific cell type, what happens if we knock it down and prevent its ability to do that? So in combination with activating the A and L genes, they introduced P53 shRNA, which knocked down P53 in the fibroblasts before it went through the conversion process. And what they found is that when they did that, the rates went from about 18% before to now just over 25%. So that's improving. We now know that knocking down P53 can help convert more fibroblasts into neurons, which is good. The other thing they looked into is something called miRNA 124. What that is a non-coding RNA that's a part of the neural differentiation process. What they found is that when they used that with ANL alone, it didn't really make much of a difference. But when they added all three of these things together, ANL activation, P53 knockdown, and miRNA 124, now over 30% of cells express TUJ1 or TH. So we now know that this combination of these three things can help convert more fibroblasts into neurons. And for the rest of the paper, if you see ANLMP, that's what it means. It's these three things being used at once. So this is good. We're making progress. We've gone from 18% before to now over 30%. But there's another problem. So here right now on the screen are two facts. First is that, like I mentioned earlier, knocking down P53 can cause the cell cycle to more rapidly advance and complete. So it'll advance into farther stages of the cell cycle quicker than normal. The other thing is that the converted neurons, the researchers figured out, had very high levels of something called EDU, which is a marker for cells that reach the S phase of the cell cycle. So they figured that these cells that were being converted were not going through the cell cycle. The cell cycle was somehow being stopped before S, and that this early exit from the cell cycle was part of the process to convert fibroblasts into neurons. So knowing these two things, can anybody see the problem that these things might have with each other? If you want to look at this graph, this can help as well. Anyone have any ideas? Ben?
Maybe the converted neurons aren't complete, so they progress too quickly and they just miss some steps.
Not quite. All right, so just think about this. P53 knocking down P53 causes the cell cycle to complete more quickly, but at the same time, knocking down P53 helps convert more cells, which are converted when the cell cycle stops. Claire?
Well, cells that advance to S phase probably have a lower efficiency of conversion to neurons. Like,
Right.
if they've advanced to S phase, they're not going to convert to neurons as efficiently.
That's right. But so the real problem here is that P53, knocking it down, is actually at the same time doing something that's both helping and hurting the process of converting the neurons. It's by knocking it down, you're making it more easy for the cells to change type, but you're also pushing cells into the S phase of the cell cycle, which they figured out makes it exponentially more difficult to convert fibroblasts into neurons. So what they figured out is it was creating sort of this contradiction here, and now it's the second problem they have to tackle. So they decided to look into, well, yes?
Can I just talk something out because I got confused previously?
Sure.
With P53 being knocked down, the cells can enter S at a faster clip.
Yes.
Correct. Right? But entering that entering S phase at a faster clip also reduces the efficiency of converting those fibroblasts to neurons.
Correct. So it's creating sort of this contradiction, I guess, because it's both knocking down P53s at the same time helping and hurting. So now they decided to look into, well, what happens if we stop the cell cycle at G1, where we could get the benefits of knocking down P53, but at the same time limit cells that advance into S and hopefully keep them in a state where it's easier to convert them. So the first way they did this was by doing something they called serum withdrawal, which is they used the ANLMP technique as they had before, except that this time, which is in this figure on the right, they grew the cells without the serum in the medium that helps them grow and survive. So basically, what this did is it effectively stopped the cell cycle at G1 and made it harder for the cells to grow. Basically what this did is, like I mentioned earlier, more rainbow is better, and there's clearly a lot more rainbow when they did the serum withdrawal technique. More cells were expressing TUJ1 and TH, meaning they've been converted into neurons when the cell cycle was stopped at G1. And in this diagram right here, they found that no cells tested positive for EDU, which I mentioned is a sign that a cell has advanced into the S phase of the cell cycle. So this is really good. We now know that stopping the cell cycle at G1 can help convert more fibroblasts into neurons. They also tried testing stopping the cell cycle at G1 through a few other techniques with the same results. So we can now add G1 arrest to ANLMP as our list of things that converts fibroblasts into neurons, which is pretty good because, like I mentioned when we started, we converted about 18% with techniques we knew before, but now with all of these things combined together, we can convert about 55 to 60% of fibroblasts into neurons. But that's still obviously not 100%, so there's still more work to be done. The next thing they realized was that they were growing cells in a medium that contains something called BFGF. And what that is a growth factor for fibroblasts. And they realized that BFGF was actually stopping, or it was doing the same thing that knocking down P53 was doing, in that it was pushing more cells into S. So they decided to replace that with NGF, which is a nerve growth factor. And what they found was that when they replaced BFGF with NGF, and then used ANLMP and serum withdrawal as they had before, the rates of conversion went from about 69% to now over 90%, just by making this one little change to what they were growing the cells in. So then they decided to look into, well, what happens if we introduce other things that help neurons grow and survive, things called neurotrophic factors. And what they found is when they did this, this is all done in the same serum medium they had before, except that they added any one of these long list of things, which are all different neurotrophic factors that help neurons grow and survive. They all individually increase the rates of fibroblasts that were converted into neurons. And then when they were all combined together at the end, it again very much increased the rates of fibroblasts converted. So now we know that adding all of these things can help convert more fibroblasts into neurons, which is good because now we've gone from 18% before to now over 90% in multiple experiments. The last thing they looked into is something called TET, which is a family of proteins that, even though this isn't their primary function, they're known for their ability to help change the type of a cell to undifferentiated, if you will. They noticed that the converted neurons had very high levels of these TET proteins. So they decided to look into, well, what happens if we knock down TET before we go through the ANLMP and serum withdrawal process as we had before? Will it make a difference? They found that yes, it did. When they knocked down any one of the 3 varieties of TET, the rates of conversion went from just over 60% to now under 10% when they knocked down TET1, which is a pretty dramatic drop-off by knocking down one protein. The rates when they knocked down TET2 and TET3 were also pretty big as well. So they inferred that TET proteins are necessary to convert fibroblasts into neurons, which is important because previously we thought that only ANL was needed to convert fibroblasts into neurons. So now we know that these other proteins are also needed. They also decided to look into adding extra TET and seeing if that would make any kind of a difference. They found that adding TET2 or TET3 really didn't make a difference, but the addition of TET1 once again dramatically increased the rates of cells that were converted into neurons. So now we can add TET1 to that long list of things that helps convert fibroblasts into neurons, which is pretty good that we now have this long list of things, considering we started here with fibroblasts and ANL activation, giving you just a couple of neurons really. But now we're here. Fibroblast, ANL activation, miRNA 124, p53 knockdown, G1 cell cycle arrest, various neurotrophic factors, and the addition of TET1 can all combine to give you lots and lots and lots of neurons, which is really good. This actually is a very big breakthrough in this field of research, really, for a number of reasons. The first is that we now have a cheap and easy way to produce dopaminergic neurons than before. And our previous methods, like I mentioned, were all expensive and inefficient. How this is important is because now we can start to investigate any potential therapies to replace lost neurons in the brain or help someone grow their own neurons, like I mentioned earlier. We don't really know quite how this would work yet, but it's something that a lot of researchers in Parkinson's have dreamed of doing for a long time, and this is a huge step forward towards that goal, which is the ultimate long-term objective of this paper. This is still something that's probably years away, but it's where this is heading. The next thing is also it can be beneficial to researchers who do research for Parkinson's because now they will be able to hopefully have a supply of these neurons they could use in a lab to do tests on them, hopefully find out why Parkinson's is caused, how we could stop it and prevent it. So these are all things that can come out of the long-term ramifications of this study. But it's not quite all sunshines and rainbows just yet. There are still, that's coming. There's still a couple problems in the way. The first, like I mentioned, is that we don't know how to take neurons and put them in a brain and just say work. We can't really do that because the brain is a very complex system of neurons and signals that we don't really understand. So we have to do obviously more research into figuring out how we would actually go about doing that. And then the other thing is, who here has taken the cancer class? Can someone tell me the statistic Mr. D always says about P53? I guess no one. Or Mr. D, you want to say it?
The P53 is indicated in about 50% of cases.
Yeah, that's exactly right. So, if you didn't hear, P53, problems with P53 are related to about 50% of all cancers. So, if you're knocking down P53 in a cell and then putting them inside a person, could that have problems? Are you giving them a cancer time bomb? We don't really know much about that. Obviously, this is something that should probably be considered if this was to one day become a therapy, how we could maybe restore function to P53, anything like that. So before I take any questions, I want to do some thank yous first. Firstly, I want to thank all these crazy idiots you see up here. This is the Journal Club team who I have the pleasure working for several hours with, helping to put this all on. You guys have been awesome to work with over the past year or so since I've been on the team. And thank you so much for being awesome to work with and helping run Journal Club. I also want to give quick thank yous to Dr. Vasilio, Ms. LaJerfo, Dr. Kirkhart for all helping me with various research, science, Journal Club related things over the past few months. Catherine Mathias, who when I first joined the team sort of helped me figure out what I was doing and how to be a functioning member of the team. I want to thank my family, obviously, for being supportive of me and Journal Club, especially my mom, who was, until I was able to drive, willing to get me here at obscene hours to come learn about science in the mornings. And then lastly, I want to give a quick thank you to all of you guys. Sometimes I'll be standing by the door in the mornings taking tallies of how many people were coming in, and it's always really amazing to me the amount of people who are willing to come in before school to learn about science they may not even understand. So I want to thank all of you guys for coming every week, being supportive of what we do here at Journal Club, and making Journal Club what it is. So thank you. Does anyone have any questions about anything I talked about? Jenny.
So TET 1 helps, but TET 2 and 3 don't help?
Yeah. So they found that knocking down TET 2 and TET 3 decreased the rates of conversion, but adding extra didn't really make a difference. Dr. Kirkhart?
Yeah, so with very, very few exceptions, adult neurons don't usually divide. So do you think that if you have made this fully differentiated dopaminergic neuron, that having unexpressed or misexpressed P53 would be enough to maybe cause division?
I mean, that is a possibility. Like I mentioned, we don't really know much about the problems that could cause. So it is possible that knocking down P53 in a neuron that doesn't divide as easily as other cells do wouldn't make a difference, but we don't really know. So it could be a problem, it might not be, but that's why we have to do more research into it. Anyone else?